good evening. This is Dr Sameeksha doing MD Microbiology in BMC. Today I will be teaching you about HIV, which causes AIDS. So, why we should know about HIV or AIDS? Okay, coming to the numerology. So, in the world, around 37.7 million people are living with HIV today, and in 2020 there were 1.5 million new cases. Globally, the prevalence of HIV is 0.7%, whereas it is 0.22% in India. So in this scenario, it is very essential for us to know how HIV infection is caused, what are the clinical features the patients come with, what are the ways to diagnose them, the best ways to diagnose them, and also treat them, and in case of needle prick injury, how to give post-exposure prophylaxis. All this we have to know. Now, coming to the structure. HIV virus is having outer envelope, nucleocapsid and inner genome. In outer envelope, there are two proteins, very important, that is GP120 and GP41. The projecting ones are GP120 and the protein which is there inside is GP41. And the third thing that virus is having is lipid. From where did it get the lipid? Lipid is actually coming from the cell membrane of the host cell that it will infect. And now coming to the genome part, you have two positive sense single-stranded RNA. Along with that, there is reverse transcriptase enzyme. Pause. Friends, now we will continue with the pathogenesis of HIV virus. Now we got to know how the HIV virus structure is. Now how it infects our host cell. So we have to know that HIV virus specifically targets our immune system, in that too CD4 T cell. Why CD4 T cell? Because HIV virus has GP120 projection, it's a protein which will bind to CD4 receptors in the T cells. So now we will have the process of formation, multiplication of HIV virus in this video. That is, the first step will be the fusion of GP120 with the CD4 receptor. Along with that, there are co-receptors like CXCR4 and CCR5. Second step is penetration, that is, virus will release its genomic material, its RNA, reverse transcriptase into the host cell. Now the role of reverse transcriptase comes, that is the third step, where reverse transcriptase will convert the HIV RNA, the single-stranded HIV RNA, into double-stranded HIV DNA. That is when there will be entry into the nucleus of the host cell, where the HIV DNA will integrate with the host cell DNA. Further, there will be formation of HIV related proteins and HIV RNAs from this process of protein formation from the integrated DNA. Now, there will be formation of immature virion. After that, the immature virion will take up the lipid layer of the host cell to finally release a newly formed HIV virus.